Hello everyone, I'm Heino Heyman, Director of Field Metabolomic Sciences at Metabolon, and welcome to episode 3 of our Molecules to Medicine webinar series. Today we are diving into one of the most critical challenges in drug development, ensuring your preclinical findings translate to clinical outcomes. We'll explore how metabolomics provides clear, actionable biomarkers that bridge the translational gap with confidence. So in this case study that I'll be highlighting, it shows the utility of metabolomics in biomarker discovery of a rare neurological disease, working from animal models all the way to developing targeted panels for clinical studies. So juvenile CLN3 Batten disease is a rare genetic disorder that belongs to a group of progressive degenerative neurometabolic disorders. It is a lysosomal storage disorder, and it typically affects children, with the first symptoms appearing between the ages of 5 to 10. Symptoms include vision loss, seizures, dementia, motor dysfunction, and it is ultimately fatal. There are 14 different forms of this disease, and CLN3 is caused by the loss of function mutation of CLN3 gene. The molecular function of this protein is still remains poorly understood, and there's currently a lack of disease biomarkers for Batten disease in general. There's no cure, and treatment is focused on managing symptoms and providing supportive care. There's therefore a great need for sensitive biomarkers that could facilitate the faster path to regulatory approval for the use in clinical trials for new treatments. The team at Stanford sought to research using metabolomics and to determine whether individuals with the CLN3 Batten disease have different levels of specific metabolites in their blood. The study is a very comprehensive study in that it includes samples from 2 animal models of the disease as well as human patients. The investigators have developed a mini-pig model of CLN3 disease that recapitulates the most common disease symptoms observed in patients with great success, as well as they have developed a mouse model carrying the same mutation. Human patient samples for the study were obtained from a natural history study of Batten disease. Plasma as well as CSF samples were collected and were analyzed using Metabolon's Global Discovery Panel doing untargeted metabolomics. Candidate biomarkers that were discovered during this analysis are further validated in a targeted quantitative assay for clinical use. Right. So if we move to the data of the study, plasma samples from disease and wild-type pigs were analyzed at approximately 6, 24, 36, and 50 months of age. At 36 months of age, 38 metabolites were significantly upregulated in the disease group of the pig model. 4 of the most significant upregulated metabolites were glycerol phosphodiesters, which are the end product of glycerol phospholipid catabolism. Of these, glycerol phosphoinositol, or GPI, exhibited by far the greatest fold change, with more minor elevation in glycerol phosphoserine, or GPS, glycerol phosphocholine, GPC, and glycerol phosphoethanolamine, GPE. Several species were also downregulated in CLN3 samples, most notably for phospholipids with deoxyhexosenol groups. Longitudinal patterns that were observed were remarkably stable over time for each of the 4 glycerol phosphodiesters, with significant elevation of each of the 4 ages for GPI and GPE specifically. The phospholipid species, on the other hand, exhibited similar trends, but with lower fold change and greater variability. So the investigators focused on the glycosphosphodiesters for the subsequent investigation. On the right-hand side, we can also see cerebral spinal fluid samples that were tested for 36-month-old CLN3 pigs versus the wild-type pigs. And finding showed significant elevation in the CLN3 samples for GPI again, but not for GPE, GPC, or GPS. In the mouse model, the levels of these four glycosphosphodiester metabolites were also measured and showing significant increases for GPI as well as GPE, but with more variability, longitudinal variability over time. An additional investigation that was included at this point was to understand specificity of the potential biomarkers. And to do this, they examined whether these changes were specific to CLN3 disease. So serum samples from the mouse models for two other genetic forms of Batten disease were taken and analyzed, and none of the glycosphosphodiester lipid species were elevated in the serum samples for the mouse model of CLN1, as can be seen here, as well as CLN6 mutations, suggesting that GPI shifts are very specific to CLN3. A very important finding for any disease biomarker identification project. Right. So collectively, the patterns observed across the pig and mouse model and across different time points suggest that the glycosphosphodiester species may be closely linked to the CLN3 disease etiology, and could therefore be useful as a clinical biomarker for this specific disease. To investigate things further, the levels of each of the metabolites were analyzed in plasma samples for 22 phenotyped individuals with the CLN3 disease. Included in that, on top of that, they included 15 heterozygous carriers and 6 non-affected non-carriers as well. Looking at the plasma data, while plasma GPS and GPC were unchanged across the three groups, as can be seen in this diagram over here, plasma GPE levels as well as GPI levels were significantly elevated in CLN3 samples, with GPI showing exhibiting the clearest separation. It is also important to highlight that the heterozygous carriers, which is this group in the middle here, exhibited an intermediate plasma GPI elevation, suggesting a dose-response relationship with GPI levels, and that they are closely linked to the levels of functional CLN3 protein. Plasma GPI demonstrated excellent sensitivity and specificity as a biomarker, with a receiving operating characteristic or ROC area under the curve of more than 98%. Comparing CLN3 patients versus non-affected non-carrier individuals. Additionally, the levels of these four glycosphosphodiesters were measured in the CSF sample of a subset of 8 individuals with CLN3 disease, finding similar statistically significant elevation in GPI levels, as can be seen here. So, in this study, metabolomics facilitated the discovery of GPI, or glycosphosphorinositol, as a potential biomarker for CLN3 Batten disease. We saw that it's consistently elevated in blood and CSF of animal models, and that researchers have demonstrated in other studies that it's also elevated in the brain as well as other peripheral tissues. Metabolomics data is currently also being integrated with other omics data such as proteomics and transcriptomics for more insights into the primary molecular function of CLN3. As mentioned earlier, GPI is also currently being used for assay development for use in clinical trials and testing of potential therapeutics. Finally, because GPI, and this is really the exciting part for me, is because the GPI is elevated early both in animal models prior to the onset of neurodegeneration, as well as in very young patients, GPI is currently being explored as a tool for newborn screening using dried blood spots as well. So really a lot of hope here for young patients. And with that, that brings me to the end of this episode. Thank you for your time and attention today. I hope you've gained valuable insights into how metabolomics can de-risk your translational research decisions. If you're ready to incorporate metabolomics into your drug development pipeline, I encourage you to reach out to our team at Metabolon. We're here to support your success from discovery through to commercialization. Remember, this episode is part of a comprehensive Molecules to Medicine series covering the entire drug development pipeline. So depending on your specific needs and interest, you can explore other episodes at your leisure, from mechanism of action studies to patient stratification studies or even drug-drug interaction studies. Finally, we are always looking to address topics most relevant to our community. Please submit any additional areas of interest you'd like to cover that you'd like us to cover in the future Metabolon webinar series. Your input helps us to create content that truly serves your research goal. Thank you for your time.